Chapter 9 is on gene transfer and genetic engineering. This chapter is really about a lot of the cool things that bacteria can do. There's not a lot of genetic diversity with bacteria. Remember, they're not undergoing sexual reproduction. They're undergoing asexual reproduction with binary fission. So they're not combining genetic sources. So they don't have the variability like we have with humans. We talk about meiosis and introducing that genetic variability so that we're not clones of our parents and not clones of our siblings. When bacteria divide, they're going to be clones of each other. So there's not a lot of genetic diversity. So what's cool about this chapter is it talks about all of the ways that bacteria can introduce different genes and kind of scramble the genes up and get some different recombinations and some genetic diversity. So a little bit about gene transfer. In the last chapter, we talked about vertical gene transfer. That's with heredity. So passing DNA down to the next generation. The only way that we can transfer our genetics is to our offspring, to the next generation. Bacteria can do the same thing. They can actually undergo vertical gene transfer, so they can transfer their genes to the next generation. But prokaryotic cells can do something that eukaryotic cells can't do. They can undergo lateral or horizontal gene transfer. So two bacteria side by side on a plate in a lab can actually exchange genetic material. This is exchanging genetic material with somebody in the same generation. This is not something that humans can do. We can't stand next to somebody and suck up their DNA into our genome. If that were true, I'd probably stand next to Carrie Arnold all day long trying to get her smarts. But bacteria can do this. And in order to undergo lateral or horizontal gene transfer, it's going to be donated from one cell to the next. So in this chapter, we'll be talking about donors and recipients. Anytime that we are combining DNA, this is what's called recombination. So these bacteria basically become recombinants. At the end of this chapter, we're going to talk about how we can actually use that to our advantage so that we can use bacteria to make vaccines and to make medicines and things that we need. The other cool thing that bacteria can do is that they can actually take DNA in from the environment and incorporate it into their genome. This is what's called transformation. Again, we cannot do this. You can't put your hand on a tabletop that maybe has some DNA and have your body suck it up and put it into your genome. However, bacteria can do this. And the first discovery of transformation, that bacteria can actually take up fragments of DNA from their environment. And the way that I remember transformation is taking up fragments. Transformation has an F in it. Fragments begins with F. You know how I like those letter associations. There was a scientist, Frederick Griffith, in 1928 that was actually working with a couple of different strains of strep pneumonia. And he discovered that there was a smooth colony, so a smooth strain that had a capsule. And remember, capsules are that really thick glycocalyx, and that made them pathogenic. They were virulent. They could actually cause disease. Why? Because, first of all, bacteria that have capsules, they're really difficult to kill. Second of all, if they have capsules, they can stick to cells. If they can stick to cells, then they can cause damage to cells, and that means they're dangerous. The other strain that he was working with was another strain of strep pneumonia, but this was a rough colony. It did not have a capsule, so it didn't cause disease. It was not virulent. So he did a couple of experiments with these bacteria. And what he did was he had some experiments where he injected the bacteria into mice. So if you take a look at this picture up at the top, so that little mouse on the far left, this was actually inoculated with heat-killed smooth bacteria. Remember that smooth bacteria had the capsule, so it was virulent. It was dangerous. Here's the thing, though. Griffith killed it. He zapped it with heat, and that bacteria died. So when he injected it, he basically injected the mouse with dead bacteria. And as he expected, the mouse lived. With another experiment, he did not kill it. So he had the live virulent smooth bacteria, injected it, and the poor little mousey died, which he expected because he knew that this bacteria was dangerous. Then he looked at the rough strain. So he took that rough strain, which was non-virulent, injected it in the mouse, and the mouse lived. So what he did was he kind of combined the two experiments where the mice lived. So he took the heat-killed smooth bacteria with the capsule, combined it with the safe rough bacteria without the capsule, combined them, and to his surprise, the poor little mousey died. So what was actually happening? In the first experiments, both of the mice lived. Why is it that this mouse died? Well, what he found was that when that bacteria was heat killed, it allowed the cell to lyse, and some of that DNA from that bacteria escaped and was in the environment. That rough bacteria then took that DNA that had the codes to make the capsule, incorporated it into its genome, and then was basically transformed into that smooth, dangerous bacteria. So this was the first evidence of transformation, that bacteria could actually take fragments of DNA from their environment. Here's my little picture that I drew out, and we've got on the left-hand side, this is our smooth, dangerous bacteria. Looks kind of angry there. And I tried to draw a little capsule. That's a bacteria that he zapped with heat. So when that bacteria was zapped with heat, it was killed and it lysed. And so some of the DNA was in the environment. So here's our, on the right side, our rough, safe, fluffy cloud looking bacteria without a capsule. It was able to take in that DNA. So here that transforming substance is the DNA. And now that safe bacteria was transformed to that dangerous, the bacteria that had the smooth capsule, because now it had the genes to make that capsule. I kind of think of this as being like the hero is transformed into the villain. And in our house, we like to watch a lot of Marvel movies. And so we always see some strange things like that happening in Marvel movies. So I think about the nice hero is then being transformed into the villain. Now, not all bacteria can do this. Bacteria have to be competent, meaning that they have to be able to open up, take in that DNA, and then close up and remain healthy. So bacteria that are competent, they actually have this competence factor. And so this competence factor, these proteins allow them to open up, take in that DNA, and remain healthy. Not all bacteria are able to do this. We could actually buy competent bacteria. So we could go to a supply house and we could actually buy competent bacteria so that if we wanted to do some genetic experiments, we know that this bacteria would be able to open up. We would be able to put that DNA into that bacteria and we could do some experiments. So not all bacteria can do this. Only competent bacteria can do this. So here's another little picture of transformation. So here we've got the bacteria, and you can see that it's got its circular chromosome, and in its environment is fragments of DNA. So this bacteria, it's competent. It can open up, it can take in that DNA, and then if you see in that third picture, it can incorporate that DNA into its genome. So it's becoming that recombinant. Recombination is happening. So in that bottom picture, you can see where this competent bacteria has acquired the genes A and B from the environment. Now, if let's say that A and B help to code for a capsule, or maybe helps that bacteria be resistant to an antibiotic, now that bacteria has acquired additional genes. So this is how one way that bacteria can actually increase their genetic diversity.